Hi everyone, my name is Emma Parker, and the article that I chose is Cannabidol Induces Programmed Cell Death in Breast Cancer Cells by Coordinating the Crosstalk between Apoptosis and Autophagy. The journal that this article was published in is Molecular Cancer Therapeutics. It was published on May 12, 2011. The listed authors are Ashutosh Shrivastava from the National Taiwan University in Taipei, Paula M. Kozuntovsky from Beth Israel Deaconess Medical Center in Boston, and Jerome E. Grugman from Harvard Medical School. And this research was supported by charitable funds for the Division of Experimental Medicine. Some introduction and background about the article is that breast cancer is the second leading cause of cancer-related death in women in the United States. That's why this article is so important. And cannabidol is a natural compound found in cannabis sativa with reported activity in breast cancer cell lines that insinuate anti-neoplastic properties. And programmed cell death is cell suicide that is critical to development and tissue homeostasis. And the two types that we will talk about in this study is apoptosis and autophagy. The main question that the authors ask is, how does CBD induce apoptosis and autophagy in breast cancer cells? And the hypothesis is that CBD induces programmed cell death independent of cannabinoid and valenoid receptor activation. For the methods of this article, they did a lot of different tests. So the first one is a cell viability assay, where cells were treated with increasing concentrations of CBD, and they were pre-treated with varying cannabinoid receptor antagonists to analyze the effects of cannabinoid inhibitors on the CBD treatment. Next, they did an apoptosis assay, where cells were treated with increasing concentrations of CBD, and then apoptosis was quantified. Next, they used an electron microscope, and cells were treated, and then fixed, embedded, sectioned, mounted, and stained, and the specimens were photographed. Next, they did analysis of mitochondrial membrane transition. The mitochondrial membrane potential MMP was used to study the changes in MMP, where cells were treated with CBD and incubated and then trypsinized, and then they used flow cytometry. Next, they did isolating mitochondrial and cytosolic cellular fractions, and mitochondrial and cytosolic fractions were isolated using a fractioning kit, and the antibodies were used to assess mitochondrial fraction purity. The last two tests that they ran were Western blot analysis and immunoprecipitation. In the Western blot analysis, cells were treated with CBD in varying amounts, and then a traditional Western blot was formed using specific primary antibodies and then incubated with their respective secondary antibodies. And the immunoprecipitation, the cells were lysed on ice and a cell lysis buffer, and the samples were gently rocked overnight with an antigen, and then the antigen antibody complexes were extracted. For the results, it showed that CBD induces concentration-dependent cell death in breast cancer cell lines in a receptor-independent manner. So basically that means that the greater concentration of CBD, the greater chance that the cell viability would decrease, and it's also receptor independent. So in both estrogen positive and estrogen negative receptor cell lines, the viability was dependent on the concentration of CBD. Next, CBD induces both apoptosis and autophagy in breast cancer cell lines. This is proved with electron microscopy, and to prove the coexistence of both, the Western blot examined the expression of apoptosis-specific and autophagy-specific proteins. Next, increasing concentrations of CBD resulted in increasing apoptosis and autophagy in breast cancer through flow cytometry. Both apoptosis and autophagy proved to be concentration dependent. Here's a chart showing a viability assay that proved concentration dependence for cell survival. And then here's another chart that shows concentration dependence for the percentage of apoptosis. And then here's a chart that shows that CBD mediates autophagy and apoptosis by inducing endoplasmic reticulum stress in breast cancer cells through inhibiting multiple different signaling pathways. And then using a Western blot, they assessed whether CBD can cause endoplasmic reticulum stress in breast cancer cells. And then it shows the levels of acridine orange for autophagy and annexin V for apoptosis to prove that CBD induces ER stress and downregulations of AKT signaling. For the continued results, CBD induces apoptosis in breast cancer cells via the mitochondrial-mediated signaling pathway. CBD induced mitochondrial-mediated apoptosis as a result of the convergence of intrinsic and extrinsic death signaling stimuli. ROS generation is required for CBD-induced apoptosis and autophagy in breast cancer cells. After incubating the cells, they observed a significant increase in ROS, and then they found that inhibiting ROS generation with TOC attenuated a significant portion of CBD-induced apoptosis, confirming a pivotal role for ROS in CBD-induced PCD. Continuing on with the results, caspase and autophagy inhibitors altered the levels of CBD-induced apoptosis in breast cancer cells. A general caspase inhibitor was used, which resulted in a small decrease of apoptosis in cells pre-treated with it before CBD incubation versus those incubated with CBD alone. BAF, which suppresses autophagy, showed approximately 15% increase in apoptosis when used alongside CBD. This proves that apoptosis levels increase when autophagy is inhibited. CBD induced molecular crosstalk during autophagy and apoptosis in breast cancer cells, which means at higher concentrations, breast cancer cells favor apoptotic cell death, and a decreased association between two receptors marks an intersection between CBD-induced autophagic and apoptotic programmed cell death. So for the discussion, the authors sought to identify novel agents for breast cancer treatment through CBD, known to have anti-cancer properties already. The molecular mechanisms by which CBD induces programmed cell death in breast cancer and the relationship between apoptosis, autophagy, and ROS generation were examined. CBD was found to induce programmed cell death independently from the CB1, CB2, and valenoid receptors. The induction of endoplasmic reticulum stress by CBD can trigger autophagic cell death through the inability to maintain homeostasis, resulting in cytodestruction and programmed cell death. ROS generation plays a key role in CBD-induced programmed cell death in breast cancer cells. Through the CRAP test, I found that the article was very credible. The article was published in 2011, but in terms of relevance, the article is still sourced today by scientists. The authors are linked linked to credible universities. The accuracy of the article seemed very precise, and the methods through which they got their results was unbiased. The purpose of the information was very pertinent due to the number of women affected by breast cancer every year. And then here's the source of the article. Thank you.